Here are the clicker questions for Chapter 6, RNA Synthesis and Processing. Use these as a general tool to see how well you know the material for Chapter 6. And as best possible, I will try and go over any incorrect answers that are listed on the multiple choice, or explain any true-false statements that are false and explain why they could be true. And hopefully this will give you a good idea of what to study for the exam. 1. E. coli RNA polymerase differs from E. coli DNA polymerase in that A. It reads the template DNA strand in the 3' to 5' direction. B. It does not need a primer sequence to initiate reading. C. It is a multi-protein complex, whereas DNA polymerase is a single enzyme. Or D. The double-stranded DNA gene does not need to be unwound for reading. So the correct answer is B. It does not need a primer sequence to initiate reading. So very simply, looking at the other answers here, again, going as if they're true-false statements, we know obviously that the both RNA and DNA polymerase is going to go in the 5', 3' direction, so it cannot be A. Whereas letter C, both the DNA polymerase and RNA polymerase are both multi-protein complexes. They're not a single enzyme. And of course, we definitely need the DNA to be unwound in order to have the actual transcription be completed. Number 2, true-false. The promoter sequence is transcribed first, followed by the DNA of the gene. And this would be false. Obviously, the promoter sequence is not transcribed at all. Only the DNA that's regarding to the actual gene is going to be transcribed into the mRNA. Number 3, the sigma subunit is of the proper size to A. Unwind double-stranded DNA of the gene. B. Bind to and cause the crab claw-like beta and beta prime subunits to close. C. Span between and bind to both the -35 and -10 promoter elements. Or D. Span between the promoter and the open reading frame of the gene. The answer is C. Span between and bind to both -35 and -10 promoter elements. So this is textbook for the sigma subunit. Otherwise, the other statements placed here, it does not help unwind the double-stranded DNA. It's for a different enzyme. It also does not bind and cause the crab claw-like beta and beta prime subunits to close, and also is not responsible for spanning the promoter and the open reading frame of the gene, because it's going to span between -35 and -10 promoter elements. Number 4, true-false. The most common way to terminate transcription is by tying a knot at the end of the mRNA molecule. A. True. GC rich invert repeats from a stable loop at the end. Or B. False. mRNA is a linear molecule and cannot fold back on itself. Of course, this one is true. So very simply, to terminate the transcription, ties the knot or the lariat position that we see in the mRNA molecule. So as we know, it definitely does fold back on itself. Number 5, chloroplast RNA polymerases are similar to bacterial RNA polymerases because A. Chloroplasts are found only in plants, which have a simpler genetic machinery. B. Chloroplasts, like bacteria, are not as large as eukaryotic cells. C. Chloroplasts perform only photosynthesis. Eukaryotic cells perform all other activities. Or D. Chloroplasts arose in plants via endosymbiosis. The answer is letter D. Chloroplasts arose in plants via endosymbiosis. So we know obviously with chloroplasts and mitochondria, these were thought to be bacteria that infiltrated other cells and basically had created a symbiotic relationship within the cell. First the mitochondria, and then second the chloroplasts. The other answers here, again going by true-false statements. As we know, chloroplasts are simply just as complex as other pieces of genetic machinery within the cell. We know the chloroplasts, we know already are not as large as eukaryotic cells, but it's a nonsense statement. And of course, the chloroplasts perform only photosynthesis, whereas eukaryotic cells perform all other activities. This does not very simply factor into any kind of differences or similarities with different RNA polymerases. Number 6, a major difference between prokaryotic sigma factor and eukaryotic transcription factors is that A. Eukaryotic transcription factors bind upstream of the coding region. Sigma factor binds downstream of the coding region. Or B. Prokaryotic gene expression can proceed without sigma factor. Eukaryotic transcription factors are required for gene expression. Letter C. Prokaryotic sigma factor binds directly to the polymerase enzyme. Transcription factors bind to DNA and recruit the polymerase. Or D. Eukaryotic transcription factors are required only to initiate transcription. Prokaryotic sigma factors are needed throughout transcription. The answer is C. Prokaryotic signaling factor binds directly to the polymerase enzyme, while transcription factors bind to the DNA and recruit the polymerase. So to go through the other statements through here, letter A, eukaryotic transcription factors bind upstream is simply false, and signaling factor binds downstream is also false, it's simply reversed. For letter B, prokaryotic gene expression can proceed without signaling factor. That's simply, it can happen, but it's very unlikely that it will bind and proceed without signaling factor. We know the transcription factors are required for gene expression. And letter D, it's a little bit more, excuse me, a little bit more of nuanced than what the other answers are. Very simply, it is false, where the eukaryotic transcription factors are required only to initiate transcription. The prokaryotic signaling factors are needed throughout transcription. That's false. Once it starts to actually transcribe the first 10 bases, the signaling factor will simply disperse from the actual DNA strand. Number 7, what would happen if a yeast cell could not make TFIIE? A, TFIID would not be recruited to the initiation complex. TFIIB or TFIIH would not be recruited to the initiation complex. Or letter D, transcription could not proceed. And the answers for number 7 would be C and D. So obviously what you want to do is follow the progression of the TFII parts of the protein. So we'll start with TFIID, followed by TFIIB, which will follow with TFIIF, and then E, and also through H. So TFIIE is no longer available, then TFIH, next one down the line, would not be available, so therefore transcription would not proceed. TFIID and TFIIB will continue to start with the initiation complex, but will stop at TFIIE. And piggybacking on the last question, number 8, what would happen if a yeast cell could not make TFIIB? A, TFIIF would not be recruited to the initiation complex, or B, TFIIE, or C, TFIIH would not be recruited to the initiation complex, and again, D, transcription could not proceed. And to expand on the last question, all of the above would be the answers for this question for number 8. Since TFIIB would be the initial part of the subunit that will start the initiation complex, therefore neither TFIF, TFIE, or TFIH would be able to be recruited for the initiation complex, and therefore transcription could not proceed. Number 9, what is particularly unique about RNase P? A, it is capable of initiating translation of both rRNA and tRNA genes. B, it is capable of binding both RNA and DNA molecules. C, it is composed of a combination of RNA and proteins. Or D, it contains an enzymatic RNA. Now, the best answer for this particular question is letter D. It contains an enzymatic RNA. So technically, an RNase P is also going to be composed of a combination of RNA and proteins, very similar to, but not exactly the same as small nuclear RNAs or SNO-RNPs. But the most unique composition for RNase P is that it contains an enzymatic RNA. In fact, it's a textbook for enzymatic RNA. Letters A and B simply are not functions of RNase P. So in this one, I think if I was grading this, I think it would take either letter C or D as an appropriate answer. Number 10, in order for a molecule of RNA to be self-splicing, it must A, be bound to a spliceosome containing at least one snRNA, B, be part of a larger multi-protein splicing assembly, C, contain an intron with ribozyme activity, or D, have all of its introns removed. So the answer is C, contain an intron with ribozyme activity. So in the absence of other types of factors for proteins or RNA factors for splicing, as long as we have an intron that has ribozymic activity, then that particular molecule of RNA can be known to be self-splicing. So the other statements that are listed here does not have to be bound to the spliceosome because the spliceosome would be absent in this case. It's not needed to be self-splicing. Be part of a larger multi-protein splicing assembly. Again, self-splicing is done in the absence of these types of assemblies. And of course, if all the introns have been removed, letter D, then there's simply no intron that can actually perform the ribosome activity. So we're left with letter C as the correct answer. Number 11, true false. Alternative splicing is one of the many examples of regulation of gene expression. And of course, this is true. The other example that's shown within text includes the RNA editing, which simply will transpose different types of nucleotide bases to give us a different amino acid, which will code for a different polypeptide, and of course give a different structure and possible different function of a particular protein. True false again, number 12. RNA editing changes individual codons. This can either alter a protein's amino acid composition or result in a much shorter protein.
And of course, this answer is true. Of course, RNA editing, this is going to be a textbook definition, is going to alter amino acid composition, which of course can give us a shorter protein.
Last one, number 13. Mammalian mRNAs have different lifetimes, ranging from less than 30 minutes to approximately 20 hours. This is a form of regulation of gene expression. One would expect the shorter-lived mRNAs to code for A, regulatory proteins, B, structural proteins, C, proteins involved in basic metabolism, or D, transcription factors.
The answer to number 13 is twofold. We have both regulatory proteins and transcription factors. So basically you want these two types of mechanisms to basically be limited in their scope as far as how long they'll be regulated for. Regulatory proteins could be anything along the lines of neurotransmitters or possibly any kind of hormones, which certain times you don't want these going on for a long period of time. You want them effectively to shut off after the 20 hours is up. Same thing with the transcription factors, as we talked about before. If we see too much expression of a certain type of gene, certain types of mechanisms can run amok. In this case, in cancer cells, can definitely amplify the gene expression and therefore cause a lot of different types of issues. Conversely, though, the structural proteins and proteins involved in basic metabolism, of course, these you do want to be long-lasting, since the structural proteins in particular, naturally you want them to be constantly replenished just to maintain the environment along the cell. As far as the basic metabolism, since very simply you're having both catabolism and anabolism happening in frequency back and forth, you want this also going on a regular basis.